In passive transport, we talked of diffusion, facilitated diffusion and osmosis. Now, active. This is with expenditure of energy. There are two main examples which we can take here. One is sodium potassium pumps, and two, endocytosis. We have talked of endocytosis little earlier. So, sodium potassium pump. What happens in sodium potassium pumps? In the plasma membrane, there are special proteins. These proteins are acting as pumps. So, they help in exchange of sodium and potassium. With 1 ATP spent, because it is active, energy consumption is necessary here, otherwise this transport will not take place. So, with 1 ATP spent, whatever energy is released, these proteins which are acting as pumps, they are going to pump 3 sodium ions out in exchange of 2 potassium ions. And because of this, the plasma membrane is normally electropositive on the outer side and electronegative on the inner side. So, these pumps which are sodium potassium pumps, they work with expenditure of energy. And why are they known as sodium potassium pumps? Because they help in only exchange of sodium potassium pumps primarily. Active transport can be primary as well as secondary. Active transport can be primary also and secondary also. What is meant by primary? Primary means if ATP is used or broken or energy is taken from ATP and that energy is used for exchange of substances. So, what is the source of energy? ATP, directly the energy source. Then it will be considered as primary. But if due to whatever has happened in the primary transport, if more transport takes place, then that would be secondary. Let us take an example to understand this. With ATP expenditure, sodium ions go out, potassium ions come in. This is what we saw in case of sodium potassium pump. This is an example of primary, because for these pumps to work, energy is coming directly from ATP. So, this is a primary active transport where ATP provides the energy. Now, when this sodium will come in the next stage, when these pumps, they bring sodium in, one more molecule comes along with it. Why is this sodium coming in? We said it is electropositive, so there are more positive charges outside as compared to inside. Is there a concentration gradient? What is more outside? Potassium ions, sodium ions, or you can say cations. More positive, less positive. Is there a concentration gradient? What did we say in diffusion? From higher concentration to lower concentration. So, now when sodium ions come in, it brings glucose along with it. Why is glucose coming in? Glucose is coming in due to the gradient difference. Sodium is moving from its higher concentration to its lower concentration. It is a concentration gradient, but due to this movement, glucose is coming. So, is energy spent directly from ATP in this case? No. So, this becomes an example of secondary active transport. Difference clear? In case of primary, the direct energy source is ATP, as we see in case of sodium potassium pumps. Due to this primary, what is the outcome? The outer side of the membrane is electropositive. It has more positively charged ions on the outer side as compared to inner side, which results into formation of a gradient. when these ions move from higher concentration to lower concentration. Some other substances like glucose, they accompany them. So with this, glucose will come in. Is energy directly spent for exchange of glucose? No. So that is secondary transport. Two more important terms which we need to talk about in active transport. One is symport and the other is antiport. Symport is movement of two molecules in the same direction. Two molecules or particles in same direction. And antiport is movement of two molecules or particles in opposite directions. Now, out of these two examples which we have taken here, which will be the example of symport? We want two molecules or particles to move in the same direction. This will be the example of symport. Sodium ions and glucose, they are moving in the same direction. So symport example is glucose with sodium ions. Antiport means two particles are moving in the opposite direction. This sodium and potassium, this will be antiport. So example of this is sodium ions and potassium ions. So symport and antiport, these are two types of, again, active transport movements. Primary, secondary we have talked of. Endocytosis we did in the previous section. Endocytosis is where something is taken in with the help of a cup-like structure formation. If the substance which is taken in is solid, then it is known as phagocytosis, which in common term we call cell eating. Phagocytosis was first seen or explained by Matkin-Koff. He explained the concept of phagocytosis. He also said that our body defends against bacteria by engulfing them. So the WBCs, they engulf the bacteria by the same process of phagocytosis. So it is cell eating. The substance which is taken in is a solid. If liquid is to be taken in, then it is known as pinocytosis, also known as pinocytosis or cell drinking, where liquid is taken in. So endocytosis can be phagocytosis or pinocytosis. If the substances which are to be taken in, if something is to be thrown out, it will be called exocytosis or ephagy. It is also commonly known as cell vomiting. So cell eating, cell drinking when it is to be taken in, and when it is to be thrown out in the form of undigested or the remain or waste, then it is exocytosis or ephagy. So these two, that is sodium potassium pumps and endocytotic intake, are examples of active transport. Here energy expenditure takes place. So here we are done with complete part of plasma membrane, its modifications, functions, models, plus transport across plasma membrane. Now moving forward, we will talk about other structures. I hope this is very clear to you. Hi, this is Neela Bakure again. In this particular segment, we are going to be talking about cell wall. Cell wall is outside the plasma membrane. It's an external layer outside plasma membrane. Cell wall is present in bacteria or prokaryotes. It is also present in fungi and it is also seen in plants. The composition of cell wall in these three groups of organisms is different. In case of bacteria, it is made up of peptidoglycan. Peptidoglycan is a molecule or a complex which is made up of proteins plus carbohydrate. In case of fungi, it is made up of a substance called chitin. Chitin is a polymer of N-acetyl glucosamine. It's a polymer. In plants, it is made up of cellulose. It is also a polysaccharide, which is a polysaccharide made up of glucose. So there are three organisms in which, or three categories of organisms where we find cell wall. Bacteria also have cell wall, fungi and plants also have it. Compositions are different. In plants, the cell wall is made up of two basic things, the cellulose fibers and matrix. Matrix is jelly-like fluidic substance which is made up of again two main things, hemicellulose and pectin. Hemicellulose is again a polymer of glucose, xylin. Pectin is also a polymer of galacturonic acid and glucuronic acid. So this makes the matrix part, and cellulose fibers are there. What is a cellulose fiber made up of? 6,000 glucose molecules make one cellulose molecule. About 20 to 30 cellulose molecules make one microfibril, and many microfibrils make a macrofibril. And in the cell wall, when we say it is made up of cellulose, we find micro and macrofibrils, and ultimately it is made up of glucose. So it's a polymer of glucose. So first from glucose we get cellulose. Many cellulose would give us a microfibril and many microfibrils would give us a macrofibril. If we talk of two cells, say this is one cell and I am just making one side so that we are able to see the cell wall. So this is the cell wall of this particular cell, say this is cell A. There is an adjacent cell and this is the cell wall of, the plasma membrane of cell B. Here we find that the two adjacent cells, they share the same cell wall. So between two adjacent cells, there is a common cell wall. So when this cell is considering its cell wall, this becomes a wall of this. When cell B considers its cell wall, that becomes the wall of the other cell also. If we see the structure, every cell wall has a middle part which is made up of calcium or magnesium pectate. This is called middle lamella. Middle lamella is made up of calcium or magnesium pectate. We will talk about this little later again. Now, we are talking of two cells, cell A and cell B. So, between the two there is this one middle lamella. Cell A secretes its cell wall. So, this cell wall is going to come from cell A on this side, and the cell wall, that is cellulose part that we are talking of, is going to come from cell B. Before we complete this diagram, let us see how many layers are there. So, these are the layers of cell wall. First is primary cell wall. First to be laid or first to be secreted is primary cell wall. So, on both the sides, cell A would secrete its primary layer and cell B would secrete its primary layer. Primary cell wall is elastic. It has less cellulose and more matrix. Water content in the primary cell wall is also more as compared to the secondary and tertiary. So, first to be put on either side of the middle lamella would be primary. So, these two layers are the primary cell walls. So, primary of cell A and primary of cell B. Then comes the second layer, that is secondary cell wall. Primary cell wall, as it is elastic, stretchable, it is present normally in younger cells, the cells which are still growing. Once the cell have grown to their normal size and they are mature, then the secondary wall is put or secondary wall is laid. Now, secondary wall is not continuous. There are areas where this deposition is missing. These areas are known as pits. This is also a pit and this is also a pit. Normally, the pits of two sides would be in front of each other so that exchange of substances can take place. So, these two layers which we have drawn here, this becomes the secondary wall. Secondary wall has more cellulose. It is rigid, that means non-stretchable. Reason, it is being secreted or laid in mature cells, and it has less water content also. Then, because it has to help in exchange of substances, there has to be an area where this exchange can take place. As we said, these areas are called pits. So, if you are talking about this membrane only, which is in this circle, it is known as pit membrane. So, what is pit membrane made up of? It is made up of middle lamella and primary cell wall of both the cells. Because it is elastic, permeable, substances can pass through this. Let us talk about the two types of pits which we see. One is called, these are the types of pits. One, simple pits, and two, bordered pits. What is the difference between the two? This is the middle lamella, the primary wall of this cell and primary wall of this side, and when the secondary layer is deposited, there is a gap where this deposition of secondary layer does not take place. If you can see this, the diameter of the complete pit is uniform, that is from the opening to the base, it is a tube like thing. So, if diameter is uniform, then it would be simple pit. In simple pits, what we see is uniform diameter. In case of bordered pits, the middle lamella, primary layer, primary walls, this part is the same. Now, during deposition of the secondary, the deposition takes place in a slightly different manner. Is the diameter of these pits same or uniform? You can see at the opening it is narrow and the base is wide. So, here the diameter is different, the base is wider and the opening is very narrow. So, here uniform diameter in simple pits, whereas here the base is broad but the opening is narrower, simple and bordered pits. In some cells like tracheids and all, there is one more layer which is laid, it is called tertiary cell wall. Tertiary cell wall is not a uniform layer normally, it can be in the form of patches, it can be in the form of a spiral arrangement. For spiral arrangement, suppose we make just a long cell with its primary and secondary wall, the tertiary layer is laid like this in the form of spring, or it could be in the form of irregular patches. So, this is thickening of one more layer which is tertiary. So, this, let us just draw a few tertiary thickenings here. Now, if we see the arrangement of this in cell A, the innermost layer of the cell wall is tertiary, outermost is the primary. The first to be produced is primary, the last to be produced is tertiary, and tertiary is not secreted in all the types of cells. Secondary is of multiple layers which can be written as S1 or S2, S3 and so on. So, secondary can be in multiple layers again. The difference between these layers would be the arrangement of the micro or macro. What does that mean? That suppose these are the three layers S1, S2 and S3. The fibers arranged here like this, in this case in some different angle and in some different angle. So, that would make the three layers. So, three layers of cell wall. Primary, it is elastic, stretchable, present in all plant cells. Secondary cell wall, secreted only in mature cells, and tertiary is normally secreted only in few cells depending upon if they need that extra strength and support or not. For example, tracheids. What is deposited in this tertiary? In tertiary is again cell wall, but cellulose, but this gets replaced by substances. Invariably the substance is lignin. So, whenever tertiary cell wall is laid, immediately it is deposited with lignin, and so we also call it lignification. So, simple structure, middle layer, middle lamella, then primary, then secondary and tertiary. If you go from inside the cell, the first layer which you encounter is tertiary, followed by secondary, and the outermost to the cell is primary. First to be produced is primary. Primary is present in all cells. All cells may not have tertiary wall because again it is dependent on the type of the cell. If a cell is in the form of a fruit or a structure And suppose the structure that we are talking about, say this is a fruit. So this cell which is here is the outermost cell. So this cell would have its middle lamella and inner layers, but nothing is deposited from outside because this is the outermost cell. So in this case, what is exposed is the middle lamella. In fruits and all, this middle lamella is exposed. We observe that when the fruits mature, the skin of the fruit becomes wrinkled. This is because of this middle lamella. It is made up of calcium and magnesium pectate. It starts to dissolve because it is exposed. As it starts to dissolve, the wall or the outer surface starts to become loose and wrinkled. This is only in case of outer cells where the middle lamella is exposed. So, cellulose and hemicellulose. Other than this, there are some extra substances or deposition along with the cellulose, hemicellulose and pectin. What that substance could be? It could be lignin, then it is known as lignification, which is normally seen in case of tracheids, vessels, fibers. It could be cutin, which is seen in cuticle. Normally leaf upper surface or upper epidermis is covered with a cuticle. So that is cuticle. It is known as cutinization. It can be a substance called suberin, which is found in cork. It is a water resistant substance and it is found deposited in cork. Silica can also get deposited. So, silica deposition has been reported in grasses. So, these are substances other than the main, that is cellulose and the matrix, which is hemicellulose and pectin. So, this is the cell wall, its arrangement and the 3 layers. Now, in the next section, we are going to start with the other organelles. I hope this thing has helped you understand the structure of cell wall, and as we know, it is a protective layer, plus it also helps in exchange of water only, not the substances, but yes, through the pits exchange of substances can take place.